Hi guys, in this video we're going to go over your flagella and the spread plate technique. So here we're going to go over the four types of flagella, and the names simply refer to the tail arrangements and how many they are and where they're coming from. So the first we have up is monotrichous, and here you see the red underline, and that's denoting the shorthand version. So mono in this case, and we have just one tail coming out from one side. We have amphitrochus, which has one tail at each end. Lophotrochus, which has several tails coming out at one end. And then we also have peritrochus, which has several tails coming out from all directions. And I drew this in two fashions, and that's because when I took my lab practical, I was kind of rushing, a little bit nervous, and as you can see is when you're not paying attention, you could see how possibly you could confuse peritrochus, especially if it's in this arrangement. Looks a little bit like lophotrochus. The big thing here is you'll want to look at the flagella, and if you see flagella going across the cell itself, and if you see that it's kind of uneven, like the tails here, they end in the middle of the bacteria, while these ones over here end all the way out here. That's probably a peritrochus, as opposed to lophotrochus, where you have no crossing over of flagella, and they all kind of come out to the same length. Now for your lab practical, you won't be expected to know how to stain these because you didn't stain them in lab. You just observe them as a demo in lab. So for your lab practical, all you'll be expected to know is the name of them and being able to identify which one of them it is on the slide. Next, we have the spread plate technique, and there's two ways of doing this, and you'll do both of them in the following procedures. This all starts with labeling your plate. Follow your lab professor's instructions, but basically it should have your initials, the date, and the bacteria that you used. Depending on what experiment that you're doing, you'll add some other stuff to it. Place the bacteria on the agar, and the way that you'll do that is it could be a pour method or it could be through pipetting. Now the following steps are going to assume that you're pipetting, but in one of the following techniques that you'll see later is that you only do the first two steps. So if you pipetted your bacteria onto the agar, you're going to then sterilize your hockey stick. It's the little glass rod that you'll use to spread the bacteria. That's what the hockey stick is. And the way you do that is you'll dip the hockey stick into your alcohol and you'll flame it. Just pass it through until it catches on fire. Don't let it sit there in the flame. Just a dip in the alcohol and a pass by through the flame should be enough. And while it's hot and while it's on fire, please don't put it into the alcohol container. You don't want to start a big fire. You'll take the hockey stick and you'll touch the inside of the plate and make sure that it's not too hot. Again, if you hear that sizzling sound, it's too hot. If you don't hear anything, then it's good enough to go and to proceed to step 5. And what you'll do here is you'll move back and forth with the hockey stick inside of the plate while spinning the plate, and that looks just like this below. So you'll have your hockey stick here, you'll be moving it back and forth as you're turning the plate. Now for the following procedures, just to save us some time, one of the steps that I'll say is perform the spread plate technique, and I'll say go to step 2 or go up to step 5. The first spread plate technique that we're going to go over is going to be considered the standard. And with this, you guys are going to label all your stuff. I think you guys are going to be working with somewhere around anywhere from like 10 to 20 plates. So in order to make sure that there's no confusion, make sure you guys set it up so that when you perform the experiment, you know where everything's going to go. Probably good to work in twos as someone's placing the microbes in and the other person can hand over all the material. If you have your dilutions set up, make sure you label everything 10 to the -1, 10 to the -2, so on and so forth. Next up, you're going to dilute your microbes in the way that your lab professor instructed you to. So I think you guys are using saline. Place the microbe from the first test tube into the saline, and then take that one and place it into the next tube that has saline. And you guys are just going to dilute it all the way down. After that, you'll perform the spread plate technique all the way up into step 5. So whatever plates that you guys are supposed to be spreading, you guys will take those and put them onto the agar, And like I said, with this one, you're going to go all the way to step 5. You guys are going to be using that hockey stick for this one. Like at the next lab, you'll observe your results and you'll perform calculations based off your results. And we'll get to that at the end of the video. Now the second technique is a plaque assay of the phage titer. Same thing as last time, we're going to make sure we label all our material properly because we're going to be working with a lot of plates, a lot of test tubes. This time we're going to be diluting the T4. And now the T4 is a virus. And that virus attacks only bacteria. And I hope you guys remember the name of viruses that attack only bacteria. They're called bacteriophages. Next, we're going to add the bacteria, which we will not dilute, to their own respective test tubes, followed by mixing the T4 and the bacteria together so the bacteria are all constant to make sure that we get a good lawn growth. We're going to let it sit for a while to make sure that we have the T4 bind with the bacteria, because the next step, when we add in the agar with the T4 and bacteria mix, and we should only do this when the agar is ready so that it stays liquid as long as possible. The reason why we let it sit is because the agar we need is to be as liquid as possible for the spread, but also the agar kind of fixes the bacteria into place, but allows a little bit diffusion for the virus when it lyses out of the bacteria. So make sure that we have a good binding of the virus to the bacteria before we add in the agar. Next up, we're going to perform the spread plate technique, and we're only going to go up to step 2 in this case. Afterwards, you're going to perform your calculations, and your calculations are going to be the same as the previous technique. Now, the difference between the standard and the phage are that with the standard, we're going to be looking for colonies of bacteria, while phages, we're going to be looking for plaques. What plaques are cavities or emptyings that bacteria were once at, but no longer at because they have been lysed by the phage, by the bacteriophage. And the standard, because we're looking at colonies, we're looking at bacterial growth, while with the phage, we're looking at the virus in their lytic phase, which is one of the 2 phases that they can be in. And the lytic phase is the active phase in which it says, hey, there's enough bacteria for us to multiply with, but not too few that if we start lysing them, that we will have no more hosts to live off of. When we perform our calculations, we have CFUs, which are colony forming units per milliliter, while with the phage we have PFUs per milliliter, or plaque forming units. And with these calculations, it can be done with PFUs per mil or CFUs per mil. In the example that I'm giving you guys, I'm using CFUs per mil. How this works is you're going to begin by taking an average of 2 dilutions. And as you guys performed the lab, you guys found out that for any given dilution that you prepared, you plated that dilution onto 2 plates. And the reason for that is because we're trying to figure out where we have just the right amount of bacteria or bacteriophage to calculate our original amount. We're going to get into how we determine what's too little and what's too few, but with this, we're trying to figure out a good number in order to determine our original sample size. Let's say we've counted the 2 plates for 10 to the negative 5 dilution, and we counted them at 100 and 200 respectively. The average of that comes out to be 150, and we keep that at the same dilution factor. So the average of these two would be 150 10 to the negative 5 CFUs per mil. And then we can use that number to determine the original sample size. And the way that we would do that is using the CFUs over the original volume to determine our original colony density. Now, I believe that you guys used 100 microliters with the dilution that we have here at 5. The original volume turns out to be 10 to the negative 6. So just quickly, because this isn't a math class, you guys had your 100 microliters, which turns out to be 0.1 milliliters. Multiply that by 10 to the negative 5, which gives you 10 to the negative 6. If you guys are unclear about that, shoot me an email and I'll be happy to answer you guys' question. We have our 150 up here from the average, dividing that by the original volume, 10 to the negative 6, and we get 1.5 times 10 to the 8 colony forming units per milliliter. Now we're trying to figure out, how do we know what's too little and what's too much? The way that we do that is we want to have plates that are somewhere in the range of 30 colonies to 300 colonies. And this is the same thing for plaques. We only want 30 to 300 plaques. The reason why is because anything below 30 is statistically irrelevant. There's just not enough there for us to go off of. And anything over 300 is just too much to count. So during your lab practical, the way this usually works is they'll give you several plates and they'll ask you to take the average of them, but they'll give you three different dilution factors. One will be less than 30, the other set will be over 300, and then you'll have the one in the middle. You'll say, hey, the first set, it's too few to count, and the other one, it's too many to count. But we're going to calculate this third one here. Well, I hope this was helpful for you guys. And if you guys have any questions, especially about this math stuff, feel free to shoot me an email or come by office hours, and I'll be more than happy to help you guys out. Have a good one.